for medications to get overhyped, but one that is kind of like properly hyped or not even hyped enough is Spinraza or Nusinersen, which is a splice-switching antisense oligonucleotide. Whew, say that 5 times fast. But what it does is it actually makes it so that people with spinal muscular atrophy can make a functional kind of like backup version of a protein that they have problems making. And so this protein that they have problems making, this original one, is called SMN1. And SMN1 is important for a bunch of different things, but when you have mutations in it, it causes this like muscular weakness, like progressive muscular weakness and respiratory failure. Now, thankfully for people with SMA, there's a backup copy, SMN2, and you can actually have multiple backup copies of it. But the problem is these backup copies have a mutation in them too. And all people have this with have this SMN2 copy with the mutation in it. So it's not the mutation in SMN2 that causes the disease, it's the mutation with SMN1. And normally SMN2 doesn't even play a role in anything because it's kind of like a non-functional gene. Because what happens is this mutation causes it to get processed incorrectly. And so this mutation actually isn't even in a part of the instruction manual for making the protein. It's not in one of the exons. So if you think about the instructions for making a protein as kind of like the instruction manual for making a robot, what happens is that instead of making the robot directly from that original copy of the recipe, of the blueprint of the instruction manual, the manual actually gets processed before it's passed off to the protein making machinery, the ribosomes. And so you can think about there being these regions called exons, which are kind of like the parts that actually have the steps of add this part. And then there are parts called introns, which are more like regulatory information. Now, in the process of making that copy, that messenger RNA copy, that RNA copy, what happens is that those regulatory regions that intersperse those actual steps. And so it might be like disclaimers or things like this. I don't know. In the case of your protein manuals, it kind of tells the cells how to process the things, when to make it, that sort of thing. And so these introns are going to be these regulatory regions, and normally they get cut out. But what happens is that this mutation in SMN2, and remember, this isn't the mutation that causes the disease. This is the mutation that just like inactivates the backup version. What happens is that this mutation is in one of those regulatory regions, one of those introns, but at one of the junctions where there's an exon-intron junction. And so normally what it does is it prevents the processing of the message. It prevents prevents an exon from being included. It's kind of like if you thought you were just cutting out some of the regulatory information, but you actually cut out an entire chunk, like maybe the pages of your recipe or the pages of the instruction manual got glued together and you think you're cutting out just some regulatory information, but you actually cut out one of those important steps. And without that important step included, well, now your cells make a non-functional version of this protein. And so you don't make functional SMN2. But what if you could get the cells to make SMN2? If you could do that, well then the cells don't need SMN1 because they've got this functional backup. And so Spinraza or Nusinersen, as well as risdiplam, these actually work by making it so that more of that exon is included, so that you alter the splicing pattern so you include that exon. And when you include the exon, well, now you make a protein that's just like identical to that original SMN1 that those patients are lacking. And so Spinraza works by kind of blocking the site where there's that mutation to prevent the mutation in SMN2, to prevent it from getting removed. So you kind of like unstick those pages so that it can get processed correctly in order to make the functional SMN2, and now you have this functional protein and it can compensate for the loss of SMN1. And so today I want to tell you more about how it works, as well as how similar and not so similar treatments for SMA work, some really cool biochemistry going on, and some really life-changing biochemistry. Let's start by just getting on the same page about some of the terminology. So the original blueprint for making a protein, the original kind of like instructions, are written in the form of DNA and a segment of DNA called a gene. So you have these inside of your each of your cells inside of a part inside of your cells. So there's this nucleus, which is this membrane-bound compartment, and inside of it are all of these instructions for making all these different proteins, and they're kind of collected together in these chromosomes, which are these really, really, really long strands of DNA that are wound up. And different segments of those strands of DNA have the instructions for making different proteins. But within these instructions, there are parts that are kind of regulatory information, these like introns, as well as interspersing regions between the genes. And then there are exons, which are kind of the information, the parts that actually need to go to the protein making machinery, that go to the ribosomes in order to tell the ribosomes how to make the protein. And the actual protein making is going to happen outside of the nucleus in this region, the general kind of like cellular interior called the cytoplasm. And so what happens is that in order to get that instructions to the ribosomes, which are hanging out here, your cells first make an RNA copy of this DNA gene. So you have the DNA gene, and then an RNA copy gets made from it in the process of transcription. And then that copy is going to then travel out of the nucleus and be used by the cytoplasm by the ribosomes. But before that RNA is actually exported, it actually gets processed. And so within that gene, all the regulatory information gets cut out so that the ribosomes are only given the parts that actually tell them how to make the protein. They don't need all that regulatory information that kind of tells the cells what to do, like when to make the protein, or when to like make this RNA, how to process the RNA, etc. And the ribosomes, they're just going to need the processed version. And how this gene is processed, there's a couple different things, but one of the main things is that it undergoes splicing. And splicing is going to be the process in which those introns, those regulatory information, is removed. And there's actually different ways that you can remove that regulatory information. This is called alternative splicing. And it's actually pretty cool because it allows you to create a lot of variety from the same number of genes. And so our cells rely on alternative splicing in order to like minimize how many actual genes we have to have and make different versions of the same protein, say in different cells. Unfortunately, mutations can cause diseases or they can inactivate proteins by causing differences in the alternative splicing, so that certain exons are skipped over, or introns can be included when they should be cut out, and things like this. Now, what happens in the case of spinal muscular atrophy is that there's actually a mutation in a backup version of this main protein. And I call it a backup version, but really what it is it's just a non-functional paralog. And so we use this term paralog when we talk about a gene that has arisen from like gene duplication. So basically in this original, there was an original version of some protein of some gene. And then that gene, in the process of evolution, somehow it got copied. And when it got copied, now you have two copies. And sometimes what happens is these two copies can kind of diverge, and they can be, this can lead to the making of proteins with new functions and things like this, which can be really cool. But other times, the paralog kind of just gets deactivated. And this is what happens in the case of SMN. So the SMN1 protein is kind of like the main protein. This is the one that's normally functional, and the one that patients with SMA have mutations in that cause them to make non-functional SMN1. And then that causes all these problems. SMN2 is a paralog of SMN1 that typically it doesn't even really play a role. Because what happens is that there's a mutation in SMN2. It's actually just a single mutation. It's single letter change in that DNA recipe. And it's in one of these regulatory regions, one of these introns, at one of the kind of like the junctions between the intron and the exon. And what happens is that this mutation, that like everybody has, it's not a disease-causing mutation. It's just inactivates this kind of like backup copy. And what's going to happen is that this mutation causes this exon 7 to get removed. So when you see a diagram like this, typically so basically all of these filled squares, these are going to be the exons. Those are the parts that the ribosome needs. These are the parts that we want to get pieced together in this splicing process to be used to make the protein. And we want this gray stuff, this introns, to get removed. But what happens is that because of a mutation in this SMN2 protein instructions, now this exon 7, it kind of gets removed when you're removing these introns. And so instead of just removing this intron and removing this intron, you actually remove this exon 7. We can use this notation of this delta sign, where this delta sign indicates that something is missing. And so in this case, we're missing the 7th exon, so we call this SMN2 delta 7. And this makes a non-functional version of the protein that gets degraded. And so typically in your cells, only about 10% of SMN2 actually gets made correctly. So it actually includes exon 7, because most of the time the exon 7 is going to get skipped over. What Nusinersen, Spinraza, and Evrysdi do is they kind of make it so that it's more likely that it'll get processed correctly. So they are able to kind of alter this site where that mutation is in order to promote the inclusion of exon 7. So they can kind of tell the splicing machinery, hey, you need to cut here. The splicing machinery is called the spliceosome, and basically it gets kind of directed to different sites where it should kind of cut and paste things together. And it gets directed to these sites through combinations of cis and trans acting factors. So a cis factor, basically this is cis same, so this is something on the same thing as the thing it's working on. Okay, that was awkward. But basically, if you have a cis-acting factor in an RNA, it means that there's something in the RNA itself that's actually having the regulatory impact. And so in the case of splicing, a cis-acting factor would be like a sequence in the RNA that, say, makes it fold up in a way that hides a splice site. Similarly, you can have transacting factors. And so transacting factors, so trans is like across from. And so basically these are outside things that come in, such as proteins that can come and promote the inclusion, promote the recruitment of the spliceosome, or kind of keep the spliceosome away. In SMN2, in both the gene and then the immature RNA copy that's made from it before all that processing, in the intron between exon 7 and 8, so this is a part that we want to remove, there's a sequence called an intronic splicing silencer, or ISS. And so this ISSN1 is this sequence in this RNA that provides a binding site for a splicing repressor. So the splicing repressor protein can actually come and bind to the sequence, and it kind of hides the splice site from the spliceosome. And if the spliceosome can't see it, it's just going to cut around it. And so this is like when we're talking about having your pages of the instruction manual kind of stuck together, so it doesn't see the splice site, and it removes exon 7 when it's and splices together these 6 and 8. Now, what happens in the case of Spinraza is actually going to bind to this site and cause it to be visible to the spliceosome, so that the spliceosome cuts there and the exon 7 gets included. Spinraza is able to do this because of sequence complementarity. So basically, when you have DNA or you have RNA, you can have it be double-stranded, where one sequence is going to serve as kind of like a template for making a second sequence due to this sort of one-to-one base pairing. So you don't have time to go into here, but you can think of it as kind of like having a zipper where you have two different halves of the zipper, and so you can have these two strands stuck together, and then if you take them apart, you can use one strand to make a copy of the other strand. Bottom line, if you know the sequence of one copy, you can know the sequence of the other copy. And if you can have a sequence that is going to specifically bind to another DNA or RNA sequence. And we call this version that can bind to it the complementary strands. And so this is what we have here in the case of Nusinersen. We have a sequence that is complementary to that ISS site. And we call about this like reverse complementarity and stuff because of the directionality of DNA. And I'm not even going to get into that here. But basically, we have the sequence that is going to bind specifically to the sequence at that splice site. So it's not just going to bind anywhere in the RNA, it's going to bind at that specific site. We call it antisense because basically when you have strands of DNA or you have strands of RNA, the sense strand is going to be the one that actually has the instructions for making the protein. And so it's that sense strand that is actually going to have a copy of that sense strand be used by the ribosomes. And so this is antisense. So it's going to bind to that sense strand and block the sense strand. So that's just where the terminology comes from. Now, oligonucleotide, basically that means oligo is like several, and so this is several. It's a kind of like a short nucleic acid chain. When we think about nucleic acids, we usually think about DNA and RNA. In the case of Nusinersen and some other oligonucleotides in like clinics, these are actually modified oligonucleotides, and they're modified so that they're more stable. The RNA in our body, say, is made to be temporary. And so those copies that the ribosomes use, those can actually be degraded, and they're supposed to be degraded so they don't kind of like last forever. And your cells have ways to degrade them on demand, things like nucleases, which can kind of like chew up the RNA or the DNA, as well as they can kind of just degrade over time because, well, it's a harsh environment inside of your body and inside of your cells. And when we're talking about these drugs, we not only have to get them to survive inside of the cells, but they also have to make it there. So we need to make these nucleotides more stable. We don't want to really mess with those bases because that's what's going to be causing the pairing to that sequence. But we can alter the backbone. And when we talk about the backbone of nucleic acids, typically they have these phosphodiester linkages where you have these like phosphate groups. So you have a phosphorus surrounded by oxygens. In the case of Spinraza, it has these phosphothiorate or PS linkages, where basically one of the oxygens gets replaced by a sulfur. And also at the 2' position where RNA has an OH and DNA doesn't have anything. So this is the position where DNA and RNA vary in terms of the base, or in terms of the outside of the base. It's where you get the deoxyribonucleic acid. That's where you get the deoxy from in DNA, as opposed to RNA ribonucleic acid. In Spinraza, it actually has this methoxy ethyl here. So that 2' OH, it actually, in RNA, it actually makes it less stable than the DNA. And with Spinraza, you have an even more stable version than there was just the DNA. Yeah, this like methoxy ethyl. And these various modifications will make it more stable and help it get through, get into and survive the path to where it needs to go, be less immune reactive and things like this. So it's going to make it more stable and bioavailable. Still, it has to be injected, and so it's given intrathecally, so through like spinal injections. But it is a life-saving treatment. It is not the only treatment, though. And so there's another treatment that's a splice alter, and then another treatment that is going to act totally differently. So this other splice altering treatment is called Evrysdi, and it's actually orally deliverable. So you take it as a pill, but it has to be taken every day. So this is the structure of risdiplam. These are some structures of close analogs, so similar versions that were used to kind of study how it works. This is a nice kind of review article that goes over things. But basically, it's going to bind to the sequence as well. But in this case, you can see that instead of binding based on like sequence complementarity, this does not look anything like Spinraza. It's actually just like this small molecule that they found that happens to bind there. And when it binds to this region, basically what it does is it's going to kind of act as a glue that can cause an activating protein a splicing activating protein to kind of bind more stably to this site. So it's going to kind of help recruit the spliceosome in order to come to this site and process it. And it's also going to kind of change the conformation, so change the shape to help promote binding and make the site more visible. So I recommend you read this article if you want to know more details. But in both the case of Spinraza and Evrysdi, you're increasing the amount of SMN2 that gets made. So these are not gene editing techniques. They're not messing with the original DNA. They're only altering the RNA. And they're altering the RNA in a way that they're actually getting removed during the processing. So each time your cell wants to make SMN2, this drug, so whether Spinraza or Evrysdi, these are going to have to be around in order to bind, in order to get this transcript to be processed correctly. And so you need this drug to always be around, which is bad, but you don't have to worry about it messing with the original version, with the original recipe, or even being around when the ribosomes go to use it. And so those are some positive things about it. But if you want something that's going to be longer lasting, then you want something that would actually potentially alter the DNA itself or provide a whole additional copy. So if we talk about altering the DNA itself, then we're talking about gene editing. And there aren't any, at least commercially available gene editing treatments for SMA, but there is a gene therapy. And so when we talk about gene therapy, we're often talking about delivering a recipe for making something. And in the case of Zolgensma, it uses a harmless virus. It uses an adeno-associated virus to deliver a copy of the SMN1 gene so patients can make SMN1. So SMN1, that's the protein that they have a mutation with. And with Zolgensma, it basically just provides another version of it. And so it uses a harmless virus, this adeno-associated virus, AAV9, as a vector or a vehicle for getting a normal copy of SMN1 into the recipe into the cells, and then the cells can make it. And so it's approved for SMA patients under 2 years of age, and is given as a single but very expensive IV infusion. I should note too that because we're dealing with the nervous system, the cells in this system divide a lot less frequently, or potentially not even at all. And so basically these treatments, if you have one of these treatments, it doesn't have to get passed down from cell to cell to cell. It just has to get into those original cells. And so these treatments can be longer lasting as long as you're able to get them where they need to go in the first place. Okay, one last note about SMA is that it is a recessive, it's autosomal recessive, which means autosome basically refers to it not being it's not being sex linked. So basically it's not on one of the X or Y chromosomes, it's on one of your other normal chromosomes. And so we call these autosomal. And basically, so remember, the chromosomes are going to be those collections of all those recipes. And you get one copy of each chromosome from each biological parent. And so we're going to have 2 copies of each of the genes. And you can, when you have a mutation in one of these, you often still have a backup where you have a functional version in the other copy. And often that one functional copy is enough to work. And enough to keep the patients from having, or the people from having, a disease. And so this is the case in SMA and other recessive diseases where you need 2 kind of faulty copies in order to cause the disease. So with patients, they have to inherit 2 faulty versions of SMN1 in order to actually have the disease. If they just have one, then they can be a carrier for it, and they can pass along, and then that's that bad gene, but then their kids would have to have another one inherited as well. So this makes it more rare than if it were like a dominant disease where you would only need one bad copy. Speaking of copies, when it comes to the SMN2 gene, people can have multiple copies of this SMN2, but all of them be non-functional. But because some of the functional version gets made even without a drug,
The more copies of SMN2 you have, the more likely you are to make a small amount of functional SMN, or like a bigger small amount of functional SMN2. And so patients with multiple copies of SMN2, like the more copies you have, typically, the less severe the disease is, or later onset and things like this. And so that can be a modifying factor as to why people may or may not develop really severe disease, or they might just develop it more early and that sort of thing. So normally this SMN2 gene, it just gets spliced really, really inefficiently. But even if you have really inefficient splicing, if you have a lot of copies of it, it can kind of hold things at bay. In the case of these treatments, though, Spinraza and Evrysdi, they kind of make it so that even if you don't have that many copies of SMN2, well, now you're still able to make a lot of SMN from it, because you can promote the proper processing of this transcript. And the proper processing is going to lead to the inclusion of this exon 7. So you can make functional SMN2 protein, which is like identical to SMN1. And so the SMN1, even if those patients can't make SMN1, they don't need it because they've got SMN2. And so this is how these splice switching treatments work, is they make it so that that gene, that exon gets included, and you make the functional full-length protein. And so they differ in how they go about this, whether it be an antisense oligonucleotide that binds based on this like a sequence, or you have this small molecule like Evrysdi that just kind of happens to bind specifically to that site. And so there's a lot of kind of complex components and shapes and things and various binding proteins. It's a very complicated site at that spliceosome, and that drug is kind of able to help glue the right things together. There's also Zolgensma, which works in a totally different way, by using a harmless virus to deliver a normal copy of SMN1 so that the patients can make the SMN1 protein based on that copy, rather than trying to get them to use the non-functional copy that they already have of SMN2. So I hope that helped you understand the treatments for SMA, as well as some things about splicing and basic biochemistry and molecular biology stuff. So hope that helped. And just a disclaimer that I'm not a doctor. Well, I am a doctor, but I'm not a medical doctor. I have a PhD in biochemistry, and this is not my like area of expertise when it comes to the actual clinical treatments. I do have a connection to this story though. So I'm not connected to any pharmaceutical company, but one of the inventors of Spinraza, Dr. Adrian Krainer, he's actually at where I did my PhD, so at Cold Spring Harbor Laboratories, and he was actually on my thesis committee, and I rotated in his lab. So Dr. Adrian Krainer is a really fantastic scientist, and I heard about this drug quite a bit, and just thought it was really exciting. And so I've done several posts on it throughout the years, and I just wanted to do a short little video. So hope it helped.